Yeah,
As
yeah, sure.
like
Yeah,
you said, like, this is more like a collaboration, and we want to know, like this first meeting, we want to gauge, right? What are our expertise, what are your expertise, and how we can, like,
yes.
make sure that we can help each other. Because yeah, as Muntaz also wants to set up a worm lab, and he could be a great resource for you. Like literally, I mean, I cannot vouch you enough how much worms are. I mean, I sound like a salesman, but worms are an amazing model system to work with, and there are no iCook protocols or anything. Very cheap, and you can grow tons. And as I said, the major advantage is the worm community is amazing. You can get mutants, you can get reagents, and everything very easily. And I see this as a great opportunity in Bangladesh, at least. I don't know how many worm labs are there in Bangladesh. I'm assuming not that many, because in India itself, there are like 10 to 12. And India is a big country, and so is Bangladesh. But I mean, I'm assuming the community is not that big, but it would be a great segue for probably Muntasir to start like that, right? Like, so
Mhm.
I'm not really sure. So yeah, I mean, great thing, like Muntasir, thanks for putting this together. So yeah, I mean, what are the other people's expertise if anybody else wants to? And thanks.
Okay. Hello, good night to everybody. Uh, I am S.K. Wazizul Islam, Professor, Department of Physiology, Biochemistry and Pharmacology. I graduated from the Chittagong Veterinary and Research University, and my basic degree is Doctor of Veterinary Medicine. And later on, I completed the MPhil in Biochemistry and Molecular Biology, and did my PhD in Internal Medicine and Molecular Biology. And during my PhD, I got experiences on the mammalian cell culture laboratory, neuronal cell line, GT17, GT1 line, and hex cells, and the in vivo experiment. I did the one experiment with the one natural products. I brought from the Bangladesh and studied in the cell line and in vivo also, and I found some effect as anti-diabetic action
Oh,
and
nice.
anti-diabetic actions, and I elucidate the pathway, mode of action, and the pathway of the extract, whole extract. Unfortunately, I could not able to separate the compound, which compound is active on the test track. This is, and the later on I did the electrophysiology, the press claim, what we call that, in my PhD uh, experiment. So it's all about in my laboratory, and we use the natural products to elucidate the antimicrobials against some organisms isolated from the poultry and livestock. And other things, we did some experiment on metabolic disorders in the animal metabolic disorders, ketosis, and mastitis, like that. uh, Another thing is that we use the enthelmentic assay from the from bioactive compounds. uh, we are growing stage, and we have the capacity to do the RT-PCR and LC, liquid chromatography, and later on we have the LCMS, and yes, next generation sequencing also will get in future, near future. I am intended to establish the mammalian cell culture laboratory here.
Mm-hmm.
I have the cell line, but little bit back. I am trying to establish this. This is my all about, and I have the some collection of the plants extract right now, and I have also collection of almost 70, 74 whole plant right now. This is all about my research and experience. Thank you.
Yeah, thank you so much.
I have a question for Dr. Aziz Islam. If you don't mind. So you said that you have like about 70 different natural plant extracts or natural products. You have like 70 different natural extracts?
Yeah, currently I have the 28 extracts.
Extracts.
These are ethanolic extracts, not
Okay.
ethanol extracts.
And
And I have the collection of 72 whole plant, medicinal plant.
okay.
The dry condition, not the extract whole plant.
And they're all indigenous to Bangladesh only.
Indigenous to Bangladesh.
And
I have collected from the Bangladesh.
and do you have any information whether there is some kind of overlapping with those found in India, like
Yeah. Since in Bangladesh and India is the close, the geographical location is the same almost. West Bengal, Assam is the West Bengal is Bengal, so that is common.
but
So,
yeah, but do you believe? But do you agree that there might be some of those that would be unique to the flora of Bangladesh?
Okay, yeah. Due to the difference in soil and environmental condition, there are some plants indigenous to Bangladesh. I hope I have to charge this, and I am trying to write a review on antimicrobial essay, and I found that the plant based on the Bangladesh and who is also found in the India.
so just as a follow-up question, like, have you run any biochemical analyses or like NMR mass spec on these? Because that will give you like a direct, like even if it's in India, because if your soil is different, like say for example wine, right, from different region, it's from grape, right, but based on the soil, based on the composition, environment, the polyphenolic contents and all that. So have you run those things yet, like, you know?
No.
Not yet. Okay. Yeah. Yeah, that would be a cool next step also, just to, because that's where, I mean, it could be like monopharmacology, but in my experience, most of the time it's polypharmacology when these compounds work, like, you know, but yeah, cool thing would be to check, like, you know, if you check like the chemical. Yeah, cool stuff. And these ethanolic extracts, have you also looked at what are the stability? Have you done any stability studies? Because I don't know how the composition changes.
No, only my student is testing the stability test or chemical test, not the stability test. I
Got it.
stored the extract in the at 4 degrees Celsius.
Got it. And maybe somebody with more chemical knowledge can also tell us like how these varies. And this is like it's a knowledge exchange. So feel free, like, you know, because some things I might not have an experience, a lot of experience in some things like a chemist or like, you know, so please feel free to contribute as well. Like it's not just we are driving, like, you know, everybody can contribute, so that would be great. And that's how we know, right? Like,
okay, so let me take the privileges. This is Atiyah Rahman. So am I reachable to all?
Yeah.
Yeah. So guys, thank you so much. It's really a good chances and privilege for me to be introduced. So actually, I'm from Bangladesh, and right now I'm working in the University of Chittagong, the Department of Biochemistry and Molecular Biology. Currently, I'm the chair of this department. By the way, I've started my career in this department as a lecturer in 2004. I've been graduating from the University of Dhaka and did my PhD from United Graduate School of Agricultural Sciences, Japan. Then I got a postdoctoral fellowship in UK, Jedin, that is in Durban, South Africa. And getting back to my workstation, I started to work on alternative medicine as well as medicinal foods. So far, we have published more than 100 articles and we have explored more than 30 plants through very detailed study. And our basic focus on the diabetes and diabetes-related parameters and in vitro, in vivo, then in silico, as well as phytochemical analysis, we do usual. And our strategy is not to only focus the individual solvent extract, rather we collect the sample and do both aqueous and organic extracts. Doing wet and cold extracts, sometimes we collect the volatile walls, and interestingly, we got some very promising plants so far, and all those are in our records. And recently, we have started with working the grain synthesized natural products, and you might have some good ideas on. Hello.
Yes,
Yeah,
we
can
can
you
hear
hear
you.
me?
Yes.
Yeah.
You are audible.
Yeah. So recently we started doing some natural products, those are green synthesized, using some silver and magnesium nanoparticles. And just a couple of days before, one of my students, he got PhD and he has been awarded. Another one is an MPhil student, he has also been awarded. And 5 students, although I have total more than 20 students in my research group, PhD, MPhil, MS students, and very interestingly, one work we presented last couple of days back, and that is on neural activity of some plants, and that is green synthesized plants. And our objective was to check the effect of that green synthesized nanoparticle in some behavioral model. For example, open field test, whole cross test, elevated plasma test, Morris Archer maze test, relative mRNA expression, and some other biochemical parameters in hippocampus we checked. And particularly, we saw the expression of some mRNA that are the very much explosive, for example, BDNF, CREB, then ERK, all these genes, how they are modulated by the effect of this plant extract, I mean the green synthesized plant. So this is in brief that actually we have so far reported, and those are very comprehensive reports. We are publishing phytomedicine, we are publishing in biomedicine and pharmacotherapy, we are publishing in ethnopharmacology, we are publishing in Journal of Food Sciences and some other prestigious journals. Even recently we published in PLOS. So I think this would be very good chances to make collaboration using this nanoparticle based or green synthesized nanoparticles with this collaborative approach, as well as the other extracts so far we have collected. And right now we have maybe more than 30 crude extracts, and some other lyophilized powders. Even we currently started some animal-based some extracts. You mean the protein hydrolyzers we are using. So I think if we can, very interesting, we must share that we have GCMS spectra, we have LCMS. Some of those are already done by some phytochemical analysis. I mean, the carbon NMR, then protein NMR. Carbon NMR and protein NMR. So all this, I think, that could help more to expand our collaborative approach if we are really interested. So, yeah, I think we can share more in our follow-up discussion. Thank you so much.
Yeah, thank you so much, Professor Rahman, and that looks really exciting. So one question I had, like a follow-up, was when you're collecting these plants or plant materials, are you geotagging the plants or something? Because like, what is the ethnobotanical side of it? Like, are you getting it from markets? Are you getting directly from like, the location? Say, for example, a plant grows in some Chittagong in the mountains, right? Are you going there and getting it directly, or is it from the market? Because there could be a lot of
that's very, very important questions. Actually, we do collect ourselves from the some ethnobotanical survey. Sometimes we interact with the people, tribe people, ethnic people. For example, maybe 2 years back, before starting the COVID-19, here, 2 of my students are present here, Khalid Johani Rafi and Tanvi. We visited the site and we interacted with some traditional medicinalist and herbalist. And interestingly, we got very promising information. engraved information from those people. And based on that information, we collected plants ourselves from the sites, not from the markets. And interestingly, we started doing the standardization of the plant extracts so that the variation could be very minimal, as well as we are trying to ensure the standard condition for preserving the materials before going for the in vivo models, I mean animal models. If we are currently using the Swiss albino rats, Swiss albino mice, and Worcester albino rats, these models we have. Yeah, hope I have given you some more information. Thank you so much.
Yeah, thank you so much. That's really helpful. And just to know all these great things that you all are doing there, it's really amazing, right? I had another clarifying question just to you, Professor Rahman, was basically like when you said you were also looking at some brain activity of these extracts, or you looked at like, have you pinpointed what neurons, or is it in the books later on if you want to see what neurons, ion channels, GPCRs, because that's where the drug discovery part, or
Yeah.
I mean, you can see a phenotype, but like pinpointing or like matching it to the receptors is the main part. Is that something that you guys will try to do in the future, or?
That's great as well. Actually, currently what we are doing, our approach is that you know very well oxidative stress. This is very important issue. And alongside with some other activities, we try to find out how the tissue or organ level oxidative stress is over there. And that's why for brain, the gross oxidative stress in vitro model as well as the hippocampus. We try to collect the hippocampus and we see the superoxide dismutase catalyst and sometimes glutathione peroxidase activity. I mean the mRNA expression as well of those enzymes. But actually, particularly, we don't focus and pinpoint the very particular neurons or the ion channels. And I think that could be our next focus. And right now, in our laboratory, maybe if I get chance, I can show you some slides. In our laboratory, actually, we have some good instrumental setup, and there's very good tools for us. For example, we have HPLC, we have LCMS, we have flow cytometry, we have next generation sequencer, we have RT-PCR, and all other small instruments. So actually, this is very good for us that we can extend our collaborative approach, doing all these things to be very much pinpointing as you said.
Sounds great. And yeah, I mean, the other thing actually, I just wanted to tell you is that I was part of this European Union initiative called MedPlant, and they had like a lot of people from all of EU who were working with natural products and who were looking at not only isolating natural products. The website is not there yet, but maybe I can connect you to those folks also. That would be something great because they have vast amount of experience with like, you know, natural compound isolation, target identification, validation, and taking it literally from bench to bedside. So that would be something also I can do. But yeah, I mean, that's in the future, like, you know, if you guys are interested, I can also connect you with some of professors.
Yeah. Thank you.
Yeah, it's really cool. Thank you so much. I don't know if anybody else wants to go. Oh, yeah.
Assalamu alaikum.
Hi.
Welcome, Dr. Guha and Dr. Muntashir and all other. Very happy indeed to be here today in this enthusiastic discussion, Zoom meeting. I'm from Bangladesh Agriculture University, and I'm a professor of parasitology. I have started my career in 1999 in April as lecturer in the Department of Parasitology. And basically, I'm a DVM graduate, Doctor of Veterinary Medicine, and then I obtained my MS in parasitology, and I have garnered my PhD from Japan in the veterinary clinical sciences. I worked on the alpha-2 adrenoceptor receptors. And I had the opportunity to avail 3 postdoctoral fellowship, one in the United States, West Virginia University, in the Professor Mustafa lab, who works in the adenosine receptors. You might have known him, and Dr. Muhammad Naim. And then I worked in the University of Dundee United Kingdom in the laboratory of Professor Mark Field, the drug chemistry, the drug discovery unit with the trypanosomes, drug sensitivity. They are approaching for the drug discovery for against trypanosomiasis. And lastly, I had the opportunity to work in the laboratory in James Cook University, Cairns campus, in the laboratory of the Professor Alex Lucas, who is working on the exosome or extracellular vesicles. And he has several parasitic models like Schistosoma mansoni and Trichuris muris. And I had the opportunity to work on the Trichuris muris model. And I have explored a number of proteins, mRNA, microRNA in his lab, and published the article in the Journal of Extracellular Vesicles in 2018. And my profession is here, so I'm doing now in Bangladesh in the Department of Parasitology, and I have a team in my laboratory, and now I have 3 PhD students, and I'm the primary supervisor, and a number of master students, and mostly 7 are working with me. And now I have also my colleagues, Dr. Shirin Akhtar and Dr. Babul Chandra Rai. They will introduce themselves briefly. So this is all about my lab and about our work. Mostly our work focused on the immunoparasitology and characterization of the exosomes, that is extracellular vesicles, and drug sensitivity test, either resistance or sensitive. So one of my master students, he did MS on the tannin containing plants. He tested some ethanolic products and aqueous extracts on the Haemonchus contortus. And from then we are trying to explore the anthelmintic efficacy of the commercially available drugs and some medicinal plants. By this time, Dr. Muntashir, he contacted me from Canada in 2018, and he asked me that whether you guys are interested to work on the C. elegans. And actually, I had the just basic knowledge. C. elegans is a model parasite and it's a free living. That's okay. Later on, I have studied and I had a contact with him. And recently, after connecting with you in the conference, Alien conference this year, he called me that should we go for a project, collaborative project in C. elegans and the neuroplants, etc. Then I have explored and discussed with my colleagues in the department. And the day before yesterday, Dr. Muntashir introduced with us the various techniques, culture techniques, and the other advantages of C. elegans research and testing the biomolecules from the natural products, etc. And then we were actually highly motivated to focus or establish a lab in our department, C. elegans lab, because we have all the facilities here, lab space and other facilities. We have incubator, cell culture room. We have limited, but we can do. We have RT-PCR, mass spectrometry, HPLC, all we have. And we have a central lab where we can do the huge works like HPLC and mass spectrometry. So, and another important thing is that we have 2 important units for plant resource. One is the botanical garden in our university, and there are more than 1,800 medicinal plants. And I sent email to Dr. Muntashir today about the depository and some photographs with the medicinal plants. And we have a germplasm center. And we demand that this is the 2nd largest one germplasm center in the world after the Hawaii in the USA. Basically, we have the raw materials prepared here. And we have a good students, master students with good or satisfactory grade in their undergrad. They are enthusiastic to diverse their focus on the medicinal plant screening, the biomolecules especially. As you know that there is a drug resistance, and we have also done the levamisole and other anthelmintics drugs, like flukicidal. One of my PhD students is working on the flukicidal drug resistance. So we are exploring that there is resistance. So, you know, the organic food animal is a popular now in the world. So we are trying to explore and focus our research on the screening of the medicinal plants, identify the biomolecules through the process, and test on the either nematode or C. elegans. That's in brief our future plan and what we have discussed. So thank you all. Thank you for patient sharing. If you have any question, I'd be happy to share.
Sorry, I was muted. That sounds super exciting, and like somebody's taking the step in setting up a worm lab. That is always amazing to see this happen. And I know Muntazir can attest to that. Like, when you can help somebody, give the spark to somebody else, right? So that sounds really, and like the facilities that you have, I think are amazing. And definitely if I can help, because not only with the neuroplant project, I can also help you set up things with other assays, like, because here we are doing mostly behavioral analysis of things, but I have also done toxicological studies. I think Muntazir's lab does that also. But not only that, we validated lifespans, we were looking stuff in the areas of metabolism. So something that a lot of you, I heard, are also working in the metabolic realm and all that. So some of those assays are very well defined in worms, and I can also help you, like, you all, like, go that direction. Or so, I mean, what we can do, maybe Muntazir, I don't know what you presented to them, but maybe both of us can work on some presentation, because today I don't know, because I just wanted to gauge what the audience or people were. Maybe we can set up like a time to show them what we can do with worms, basically, right? What all assays, what subset of assays can we do, how they can leverage worm as a tool to expand their, like, whatever they're doing, right? Like, not only, like, even if, like, say for example, we cannot test the compounds here, they can do some simple basic assays there, because as I said, a lot of these things are DIY. So I mean, if you have a microscope, if you literally nowadays, even if you have a small camera, you can, like, do those imaging and all those things with the rig and all that. So it's, and I don't know, like, I don't know how the IT sector in Bangladesh is, but if you have somebody with computational skills, I mean, it's like, basically take the images and analyze them, and we can really help you with those things. And I think Muntazir could help, because these are very easy to set up systems. At least what I have worked, I have tried to make these, like, simple, easy to use, rather than, like, because you can do the same thing with $1 million equipment, but you can do, like, at least get preliminary lab setup things with, like, something in the range of, like, $100 to $200, or, like, let's say max 500, right? And I don't know, again, sorry, my ignorance, I don't know what the taka conversion is, but you can set up, like, a very cheap lab if you have, like, close to $1,000 also, like, just to get your foot wet. Or, and then, like, once you have that, like, I mean, it's like an investment, right? You make a small investment, or, like, you go to the share market, you put some money, you see, oh, it's working or not. Like, let me just see, you don't want to put, like, a crazy bit of money at the beginning. So once you have those things, a step by step, tiny steps, then could be like a big, like, thing. And definitely there's a lot of potential with worms. Like, yeah, I mean, there are, like, and I will be honest, like, there are a lot of things that you won't be able to know with worms, but think about how much you can know with worms, right? Like, how many systems you can explore, how fast your discovery rate could be, right? Like, as I was mentioning, right, with worms growing, things are super easy with doing them, like, certain assays, like, would be, like, done in matter of, like, certain hours, 2 days. And mutant collection, and I don't know, Muntazir, you can tell them also how, CGC, how easy is it to CGC or other worm labs, like, so
So
that's
one
a
thing,
very,
Yeah. Thank you so much.
and one thing I just wanted to add, and one thing, and that I also introduced, in that, basic introductory slides just a couple of days ago, is that the high throughput thing can be done only in silicons. You cannot use parasitic nematodes to do high throughput screens. So Dr. Talukdar, you mentioned that you have in your collection 1,800 medicinal plants. So how many of those have you tested? I'm pretty sure not all of them. So that you could do, I mean, in a systematic manner, in silicons. So and wait, others' help, we can, once we have the Silicon Lab up and running, not only from anthropometrics perspective, but from other like neurological perspective, anti-cancer perspective. I think you could do so many things in silicons. You need microscopes. The worm is a transparent animal, so you can see GFP expressing a protein, for example, if you have the right kind of microscopes. So, and obviously you do have those microscopes. So you just have to have silicons and its food, the OP50 strain. So back in 2018, when I came to Bangladesh, I actually did bring OP50 strain with me. But obviously, bringing the silicons is more tricky because, it survives only in a temperate, a climate. So yeah, so if we can get hold of silicons, and I think some basic like knowledge and direction, I think it can be done. So I just wanted as you give yourself as you introduce yourself to Dr. Guha, I just wanted to to remind you that you do so many different things, but at the end of the day, you have to see how you can contribute to this project that they are involved in, right? So I mean, you guys are in charge of big collections of NPs that they can maybe make use of, and you do also have data, GCMS, LCMS data that maybe they would like you to upload or share. I don't know in what ways. And if you do have NPs, then or extracts, are they in dried form or in solvent? And if there's any scope for a shipping of those materials, and how unique are those materials? Because I presumably the people in American labs and elsewhere probably have done lots of collaborations with Indian labs, and you don't want to replicate the process. You want to, maybe they have a process to de-replicate at some level. I'm not sure, but I think if you just could also help in that de-replication process, in addition to maybe sharing the or collection, maybe it would be helpful for them.
So just to add to what Muntasir said, I know at least in some of the countries, in India especially, I know nowadays this material transfer and all this knowledge transfer has been very difficult because for obvious reasons, like a lot of Western countries, like for example, basmati rice, right? Like, I know
aromatic rice. Yeah.
Exactly, like what they did is like they started to try to patent it so that like Indian growers, so there's a big barrier. I don't know how this material transfer and that with Bangladesh is there yet, because that is a big process to start with, right? Because obviously you need to have trust in both the areas, right? Like, what you are giving, there should be like, you should trust with us, like we don't take the poach on your stuff, and like, I mean, I don't know, like there's a whole bioethical issue with this, right? So we have to look into all that, right? Because that might be the biggest hurdle to start, like, things here. I mean, we do have all the facilities and all that, like, and we have some of the best brains here, but if just to get the ball rolling, like, you have to first figure out, like, these MTAs as well as like, legalities, how much flexible is the Bangladesh government or the authorities are there for you, because you cannot directly send me something, right? Again, I don't know, like I'm just saying it from my previous experiences. Sometimes it has been a hassle to get these kind of things, but depends, like, I mean, you guys, some of you might know because you probably have sent items, like, so we'll have to figure out those things first, like the legalities. But in general, I think, Muntazir, you agree, we can help them at least set up some basic labs there, like if they want to do some basic experiments, like with worms, like some basic assays. But definitely end goal would be like, if we can get the compound here, we can do a lot more. I mean, I'm not saying like, but here also, like the facilities as well as the collaborations that we have, our network might be more like, we can do more not saying like, Bangladesh does not have, like I'm pretty sure what Professor Rahman said and you guys said, the facilities I heard, it's impressive, right? Like, sometimes even good universities here don't have those. So, yeah.
Right. In response to Dr. Guha, regarding, I had a project with Wisconsin Stout University. We have an agreement in material transfer. So I did it in 2017 and 18 for testing some drugs. And our university helped. We have a protocol, set up protocol to help and to receive dispatch material. So I know already that. And also I did completed a collaboration work with James Cook University. I sent the hookworm from Bangladesh and they send some materials. So we have this protocol set up here in our university. And about the legal rights, we have procedures for patent or the sharing, other things. USA there is set up also, I know.
Yeah.
I mean, that's why. This will be easier, I think.
Got it.
Don't worry for that. Yeah.
That's amazing. Like, good to know, like, because again, for me, one time it was like some of the issues I had faced in Europe when I was back in Switzerland. And I heard that from others also. And we had to go through this big bioethics, like, seminar and all that, like, because of that.
There's a hassle, too many hassle in the
I had
airport.
an experience on that. I had sent some plasmid, structured plasmid from Bangladesh to Hong Kong, and they the authority asked me whether these are the biologically active or not, infectious materials. They asked when I shifted the plasmid to Hong Kong University. Now, we have the material transfer agreement with different universities in the UK and Singapore, like that. Samples are shifted to Singapore for analysis. Perhaps there is the opportunity to ship the product starts on the plant. I agree with the Professor Hassan Talukdar.
And one other thing, that there's a different issues when we are transferring materials from university to another foreign university. It's much easier, but the government procedure from Bangladesh, either the department or some ministry, there are much more steps, a bit difficult. But university to university, it's more easier, faster. That's
I think issue. For us, it was like a European related thing, so that's why it got more complicated. I think. Yeah. Anyways, I see somebody raise the. I think Dr. Rafi raised his hand. He has something to contribute.
Yes, it was an inquiry, a short inquiry to question to the Muntasir sir. Sir, we have know that these C. elegans are the micro devices which actually works in a single chemical stimuli for specific genes or the molecules. Sir, the question is, can we use this model C. elegans in any non-communicable disease model? Like we can use the animals for the diabetic model, cardiovascular model. Can we use such C. elegans for a specific non-communicable disease model? Is it possible, sir?
I think it's possible. It's definitely being possible. It's being done. A variety of 36% of silicons, or about 38% of silicon genes have a human homologue. A lot of human diseases have been modeled on silicons. A lot of non-communicable diseases. So you can just do a PubMed search, and you can see how many and what diseases are being modeled on silicons. Diabetes is one disease condition that is modeled to an extent, and so is a liver disease also. Also problems with like diet and fat, and how it can affect lifespan. Also stress. Also. Dr. Guha can help me, I guess.
Sure. So I can also say, like, I worked with mitochondrial diseases before I came to this position, and we were modeling mitochondrial diseases in worms. So the advantage advantage of worms is like mitochondria is the activity is highly conserved. So what you saw on those, and actually I can share one slide because I know we are running out of time. So like I'll just share one slide about the mitochondrial. Can you see my screen? Okay, let me
Yes,
put
it's
that in.
visible.
Okay, so just to show you like, so what happens, so this was the work I did at Children's Hospital of Philadelphia. So here you can see like a mitochondria. So we were working with GAS1 worm, so FC21. So it has a mutation in the mitochondrial complex 1 in the iron sulfur cluster, and it causes very debilitating diseases. Like, so for example, if you see mitochondrial patients, they have a lot of problems, they die younger, they have neurological problems, they have metabolic problems, like muscle mass is less, like pseudo problems. So what we did was we modeled that in this worm, C. elegans, obviously, and that's GAS1 FC21. And then you see, like, if you see the lifespan curves, as I said, like, lifespan can be done in 20 to 21 days. So N2 is the wild type. So the wild type worm, the average lifespan is 15 days. But in the GAS1, the average lifespan is about 11 to 12 days. So there's a significant reduction. So you model, like and you see that in patients also, right? Like they live lesser. Then if you see, I don't know if you can see this animation here, the motility is very less. So if
Yes,
you
sir.
see the into the wild type, it's moving a lot here.
Yes,
And if you see the GAS1, you see the motility is way less. So
yes,
there's that. Then if you see the mitochondrial physiology, like, you can do a lot of dyes and staining to see, like if, the mitochondrial mass is all right, or if the membrane potential, which is like a key indicator of mitochondrial function, if that's messed up, there is that. Then you can also tag these proteins very easily with GFP or other fluorescent proteins to literally see, like, what is the location of the protein, like how is the turnover and all that in a whole organism. As Muntasir mentioned, it is a transparent organism, so you can see, like, subcellular location and all that. And then also if you see the transcriptomic profiles, you can see so many dysregulations happening. The wild type is in red, and these yellows are the genes that are dysregulated in GAS1. And this is just one mutant. We have generated a series of lines, like the CLK, there's that complex 1, complex 2, complex 3, complex 4. So there are other mutants, and each and every one of them have some sort of like problems which we have looked. And then what we did was we were testing pharmacological interventions or compounds which would benefit the worms. Then what we'll do is, like, after we tested this, we'll do a further testing in zebrafish, human cell lines, and then if everything was correct, we will take it to clinic. So that is how we were doing it. In our case, we were looking at a lot of repurposing of drugs. So that was, and this is just to show you, like, just the part of non-communicable diseases, which I worked on. And then I worked on also a lot of things in endocannabinoid pharmacology, which is also, like, there are fat-related problems that happen, like fat metabolism-related problems. Then
yes.
there's also like insulin signaling, which because of worms, we know a lot.
Yes, that's
Because
great, sir.
worms
That's
were,
good. That's great.
yeah,
Thank you,
so
sir.
yeah, no worries. And I'll stop sharing my screen. But since we are running out of time,
Hi, Sujay.
yeah, yeah.
Can I ask something? Like the slide that you showed, the transcriptome profiling. So how many samples was there in the transcriptome profiling?
So, for example, when we collect worms, we collect close to like I would say like 10 to 20,000 worms, and then we do
Okay.
it in like we do 4 replicates, and then this is average of that, like 10 to 20,000 worms. So it's not individual. So you can also do single worm transcriptomics, but it's better to do when it's population, in case we do like.
No, I'm just asking just to get an idea, like how many data you were analyzing, because this comes with a cost. Transcriptomics is a little bit costly compared to proteomics, because when I used to send my samples in Purdue, proteomic samples was like around $200 per sample. But with transcriptomics, I guess it's going to be around $1,000 if I'm not making a mistake. So that's pretty high in cost.
Yeah.
But anyway, and another thing I want to ask, like, what is your expectation um, like let's say you come from like you form any collaboration, so what part you will want to do in your lab and what part you will want to be done in the collaborators lab?
Uh, so at the moment, like I still have to like I need to think about it because I have to talk to my other peers as well as like the other professors here at Stanford also,
Yeah,
because the neuroplan project, and I forgot to give you an overview, and I can give it to you the other time or next time if we meet,
yeah.
is like it is a collaboration between a bunch of labs, not only in Stanford, but Carnegie Institute also,
Hmm.
which is another institute here. And basically there are three major PIs or professors who are involved,
Hmm.
and I'm the staff scientist. I'm basically in charge of all this. So
Hmm.
they are the major people. And then there are like other Stanford
Hmm.
institutes which involve like neuroscience,
Hmm.
chemical biology, and all that.
Hmm.
So we have to sit together and then determine. But at the moment, what we're trying to look at is chemotactic behaviors.
Hmm.
So for us, it will be probably a limited library that we want to test ourselves because we are still making the platform better. So
Hmm.
for us, I think it will be best if we could test it ourselves. But
Hmm.
I might be wrong. Like, let me see, like, where that goes, because it will be a limited library. We test, like, say, for example, 200 to 250 natural extracts. We start with that as a point, and
Hmm.
then from there we go, because for you probably to test all those, I don't know if you have the facilities yet, but maybe you'll have like that.
Um, for us, like we have like all kinds of facilities for human cell culture. So
Exactly.
doing the worm work will not be hard, and for us, like we purify the compounds to the pure form from the
Mhm,
crude extract, and we do the NMR and mass spec.
mhm,
So I mean, doing the preliminary screening part will not be hard for us,
mhm.
but if you want to do transcriptomics, that involves a lot of cost. So I'm not sure the transcriptomic part can we do here. But the data analysis part, if you need any help with mass spec data, like proteomics data, that we can do. Like you can run the samples on your, I mean, on your station, and if you share the data, we can surely, I mean, we can provide support that so that you can do the expression profiling from the mass spec data, and you can say like which are like which proteins are going up and which proteins are going down and how they're affecting in the network. I mean that part we can assist you. I mean, yeah, I'm sure you can already do that with transcriptomics, but I'm just not sure if you need any if you have any plans with proteomics or source. If you want to do that, we can be helpful with that thing. And as I said that we already have capacity enough so that we can prepare the we can purify the pure compound from the crude
Mhm.
and do assays on the cell line. And if we get the C elegans from Muntasir, we can, I think we can do the C elegans also. Sounds pretty straightforward. And as I said in the first part, that we're supposed to get a patch clamp next year. So any
Mhm.
kind of ion channel screening on cell line, on oocytes or on hexel, expressing those ion channels and screening those ion channel blockers. This can we can also do. So this all depends on forming MOU or some kind of agreement, because I mean once there is an agreement, then that's official, and we can
Makes sense.
start working. Yeah,
And
that's all.
super. Yeah,
And
thank you so much.
yeah,
Sorry, you're saying something. I cut you. Sorry, so sorry.
yeah, thank you.
And yeah, I mean, that is something actually we can explore. Maybe what we can do is you guys start with probably a library of compounds where you can screen on your cell based assays. And and then we can bring it here for the worm-based, like, so the preliminary part, because I assume cell-based assays would be faster, and you have the facilities already there, right?
Yes,
So something
yes.
on those lines we can do is basically, and again, I'm just discussing this model right now. I don't know yet what is, like, so maybe, like, faster cell-based assays with certain GPCRs, ion channels, or something like that. Like, you can look into that, and then basically what you identify.
Ion channels we can do once we have the patch clamp. So that's
Yeah.
not going to happen before next year, 2022.
Okay.
Okay, but right now we do the cytotoxicity tests. That's also on cell line, and like I'm expecting like maybe in December we can get the C elegans from him, from Muntasir, because he kept those in Chittagong, and we're shifting. Our lab is shifting. Okay, and like this lab will it's going to be an institute, a whole institute. So basically the purpose is to get all kinds of facilities for pharmaceutical science research under one roof. So basically, you name anything and we'll have it, like NMR, mass spec, next gen sequencer, fluorescence microscope, like you name it and we'll have it, something like that. So
Sounds exciting.
Yes.
yeah, so like doing the stuffs here will not be an issue. For few things that may involve like some kind of high cost, like doing the transcriptomics, and this will be a matter of like because I mean because the cost is very high for transcriptomics, and that's why we prefer proteomics, because of the cost is involved in lower.
Thank you.
But anyway, I think what I wanted to say is that a big part can be done here, and that's how should be the collaborations, like win-win. Yeah, we do some part and you do some part, and that's how collaborations work. That's how I understand collaborations. I mean,
Yeah.
yeah.
Yes.
And that's, sorry, Muntasir, go ahead.
Thank you. No, I just wanted to quickly, there is one other, a professor, he has been silent. So I just wanted to ask you if he has anything to add, because now it's almost time for us to wrap up. You have given us a lot of time, Dr. Guha. So,
No.
we don't want to abuse your generosity. So just quickly, Dr. Sukulen Kumar Kundu, do you have anything to say here, Dr. Kundu?
Thank you very much. Actually, I like to thank Muntasir and also Dr. Sujoy for arranging such a nice session. And I also like to thank all other members who have joined today. Facing some problem in my internet, that's why I joined late. But from my side, actually, I'm a pharmacist. So I usually do some work based on trial, and also I have some work on the natural products also. But in comparison to Dr. Shahi, Dr. Atia, Dr. Talukdar, I think I have less facilities here. But what I found, if I like to do the research with C. elegans, it's possible to develop a lab like that. But for the time being, I think if we can establish that kind of lab in, I found 3 different part, actually, Agricultural University, then BCSIR, and the other one is Chicago University. So for the time being, I think you can just progress your work with them first, and I will be in touch. And if I like to do something, I may join with them. I can contact them, and I may work with you through them also. So it's not a big issue, but I wish such a nice protocol will be established in our country very soon, and we can do better work, better research work future that you showed really this C. elegans, the the prospect of using CLE. I think there will be good work in near future. Thank you very much.
Thank you so much. Maybe Muntasir, for next steps, as I was mentioning, what we can do, and I can rope in some other folks that I know, is what are the strengths with worm assays, because they also need to know, right? Because with NeuroPlant, our only focus is chemotaxis assays, but worms are not limited to that, right? Yes. So maybe something we can just show them, like, what are the capabilities, what kind of assays they can run, right? Like, as somebody asked, like, if there's any non-communicable diseases. So there's the metabolic aspect of it, there's the neuroscience aspect of it, there's the anti-helminthic side of it, right? So then, like, people can gauge which points, right? And maybe, as the Dr. Kundu said, right, like, there'll be a center, a nationwide center, say, for example, let's dream big. Like, if there's a Bangladesh nationwide center for worm research, maybe there could be like some people who can like pick and choose, right? Like, these are the assays to go for, right? These are the parameters that we can, like, do that. So first of all, I don't know, like, you presented them about worms, right? But did you present them anything about different aspects of worm biology where they can exploit? Because obviously, as I said, like, cancer biology, you can do stuff, right? Autophagy, actually a lot of things that we know about autophagy as well as insulin signaling is because of worms. Yes. Like, first mutants, and like cloning and all that is super easy. And yeah, I mean, the transcript, just to add to, I don't know if I'm saying your name right, Dr. Shahi Man.
Yes,
Sorry, I probably butchered it, like, Shaheen, yeah. Shaheen, sorry, I probably butchered it, my bad. But
okay.
yeah.
My name is kind of weird.
No, it's not weird, like, so what I wanted to say with transcriptomics, I do get your point, like, it's expensive, but we don't do transcriptomics also on everything. We just do it on, like, some things that is super interesting to us. The gas1 model was one of the ones that we really wanted to look forward to, and that's why we did it. Or whenever we're doing, like, say a compound that has shown us benefits in other things, and then only we'll do it, right? Like, at the end.
But as Muntasir was saying the other day, that when you want to find the mode of action for a compound, you want to find out which mutation is causing the resistance to go away. So I mean, to do that, he was saying that you guys did whole genome sequencing or something like that, right?
Yes.
Yeah. So on that part also, like doing whole genome for 20 samples is kind of costly.
Exactly.
Yeah.
So that's why you have to pick and choose of with what is your, like, for example, like, and I think Dr. Talukdar can say also, like, once you have a compound that you really feel like, is showing all the phenotypic benefits, right? Then only what you do is, like, you will go for, like, more like
that's where, like, proteomics can be helpful. Like, you do proteomics for 20 sample is just $2,000 or maybe $4,000. It's nothing. And then from there you pick your targets and you do targeted sequencing. So that will be like doing the whole thing for like $6,000 or $8,000. You save a lot by doing that.
Yeah, and I totally get your point, because why hedge? Like, don't put everything in the same basket. Like, you have to pick one thing and then from there go about, because then the great thing is that once you have one compound, what you can do after you do a transcriptomic analysis, what you can do is, like, maybe you can enrich the data and find out what are the targets. Once you have a target, that's it, right? Like, you can build, like, chemists are there who can make 20,000 molecules for the same target. And I think Muntasir's lab does that, right? Like, they are, like, good at that. So.
And also, like, there are mass spec based assays where you can shoot the compound. Sorry, you can do a binding assay with cell lysates, and then you shoot them in mass spec, and you actually can figure out, like, where the compound is binding. This is recent work I saw in a talk recently, and I have to go into the details to replicate that in my lab, but that can be done. I mean, that's
Totally.
somebody did it, so that's possible.
So yeah.
Yeah.
So,
I mean, these binding acids are very exciting. Like, you know, you can do so much. Like, as
And there are
long
many
as
ways. There are many ways. Like some guys, they do like SPR, surface plasmon resonance. Some people do mass spec, and there are like some kind of freight assay, freight assay, some kind of like fluorescence based assays, some kind of fluorescence based assays. But those are time consuming because they have to be developed, and those cannot be done like freight or bread. Those cannot be done for high throughput. So mass spec can be done with high throughput, but not the other ones. Yeah.
so, thanks, Dr. Shaheen and Dr. Guha. I know, this, I'm regardless of, what's, going to happen with neuro, the plant project. I think, if if you are willing to maybe, share some, different aspects of, like, what can be done with silicons. I give them, some like introduction about what silicons is, what can be done, and what we do in our lab. For genetic screens. So once you have a like a molecule that you're interested in, of course, you're gonna mutate the one's DNA and looking for suppressors. And then you do sequencing, and then you find, okay, maybe this is probably the target of this molecule or compound. And if you don't have the money, you can always have some inkling, maybe what kind of proteins or cell types could be the target of this molecule, and maybe order those mutant strains from TCGC if those mutant strains are viable. And then test them one by one. Test them and then see, okay, maybe this mutant shows a bit less of the phenotype. There's some kind of a partial suppression or full suppression. So maybe, so you could do all these kind of things. So my, role in all this, obviously, I'm not an NP guy, and I'm also not a mass spec, guy also. So I'm a worm person. I have been working, with worms since my undergrad days. So for more than 10 years now. So what I can do is, regardless whether there's collaboration or no collaboration, I can always, or maybe for future collaboration, I can always go back and forth between, the two continents, and facilitate exchange of data, information, and knowledge and experience, and of course open up Silicon's lab. I think since nobody has done it yet in Bangladesh, I presume that that lack of, maybe that desire was missing. So now we have it. Now I have it. So I have the expertise, I believe. So I can open it up and have it up and running. Maybe through a postdoc, if there's a nice project that I can come up with, and I do have some ideas, elegans. So that's something that maybe I just wanted to put out there on the table. So thank you. I guess if Dr. Go has something to say at the end.
So just to wrap up, I want to say like maybe like we should organize a follow-up of this, if you guys agree. So where I think a good idea would be for us to take like, it could be a 2 hour, because it looks like there's a lot to discuss and learn. And as I said, it's a learning, like we don't know a lot of things. We are presuming you want this, right? But you also need to like, know what you're getting into, or like, so that's why like you want to show you what are the capabilities of worms. And maybe like another good aspect would be if you guys can present something from your side, what are like, what are the main things that you can do, what you bring to the table. So we can present what we bring to the table. And also, as Muntasir mentioned, right, like with the purview of what projects we are doing right now, maybe a lot of things might not fit. Like maybe you're not looking in neurosciences, right? Maybe you're not looking at chemotactics, but what other things that you can do. And we would be happy to help you set up all those things, because we don't have to limit ourselves if it does not work with neuroplant or something, but you can use this whole system to do so many other things, right? It's not just like one chemosensation assay, or like, one ion channel, or one like, looking at like the ion channels or neurobiology, but metabolic sciences, as a bunch of other things, like, so we can give you that whole flavor or gamut of things that you can look and explore into. So maybe organize that, like, sometime. And I don't know, Muntazi, since you are taking the lead, like, how does that sound? Like, does that sound great to you guys? You guys present some things from your side, we get your things, and we also present some things with worms, like,
Yes. Yeah.
on screen.
Yeah, sounds good.
Yeah, yes, just to me, if sounds good, and I think since you have shown interest, so I think we should definitely go for it. I would also like to because there was so little time between the conference ending and then this thing happening, there are a couple of people I would also like to get involved from, especially from Department of Botany. We have some people from Department of Parasitology, but there are people who actually have probably can give you also more information on the state of the NP collections in Bangladesh. So that's one thing, and I think I also get involved with some people from Dhaka University, I guess, who also have some can also tell you a bit more about the state of biochemistry and microbiology, and what they can offer. So I have been in touch with them. I'm just waiting for their emails. And with regards to the C. elegans talk, I guess that's something that we can discuss in on the side. But one thing would be to just have C. elegans shipped from a C. elegans lab in India to Bangladesh, because it's a shorter distance, and I think they will probably survive that.
I mean, I used to get it from to Switzerland, and it took, like, because C. elegans are very hardy. So like, actually you can get it. You just have to set up an account in CGC if you want to. And it takes, like, the annual membership is like $40, and to get one strain is like $10. Like, so if you get more strains, it's very cheap, and it survives the whole shipping. And the shipping, literally, I was surprised that they ship it to Switzerland also. Back in the day it was $7, but now it's 10. So they were shipping it for that flat rate. So it's all funded by NIH, so they're happy to help and ship. So yeah, I mean, it will be easy. And as I said, if you ask somebody in a worm lab in India, they can, like, literally send it to you, like, easily. Even I probably can send you some wild types, but yeah, I mean, it is nothing, I think, like, even if you.
And at the end, Dr. Guha and to everyone, I would like to add that 70% of the Sundarbans actually is in Bangladesh, and because they have not been really explored, that flora and also fauna have not been tested really yet in a high screen platform. I think that whole realm is completely unexplored so far, because they're doing some test research going on on the Indian side of the forest, but it's so rich, so diverse, so we can probably avoid getting too many of the Me Too compounds, because we are now exploring in a previously unexplored geographical region. So maybe I'm pretty sure there will be many compounds, many molecules that will have really unique and distinct structures, and not necessarily like Me Too compounds. So I think with silicons at hand, I think that's something really like really doable. So I think we just have to use our own resources that we have, and it's something that we also have on the like to offer to others.
Okay. I think that's also, we went a bit over time, so I'm sorry about that. Like, we kept others here, and I think it's pretty late in right now. It's like ঘুমানোর সময় হয়ে গেছে.
It's okay. আমরা তো আছি, আপনারা তো সময় করেছেন কাজেই।
আমার আরো দুজন বাঙালি বন্ধুরা আছে যারা সি এলিগেন্স এর ফিল্ডেই আছেন, তো ওদের আমি জিনোমিক্স আর কাজ করে, মানে মিউটেন্ট আর এগুলো, আর ও বলতে পারে যে কত ইজি। আর আরেকজন হচ্ছে মেটাবলিজম ফিল্ডেই আছে, সে বাংলাদেশি, তো আমাদের সাথেই ছিল। তো হ্যাঁ, মানে এই বাঙালিদের হেল্প করলেই ভালো, রাইট?
আমি কিন্তু আমি কত বছর গেছিলাম আপনাদের কলকাতায় গেছিলাম আমার বন্ধু থেকে। আপনি নিশ্চয়ই ফ্রম কলকাতা, রাইট? কলকাতা থেকে আপনি?
আমি কলকাতা, হ্যাঁ, এখন কলকাতা, কিন্তু আমি আগে অন্য জায়গা থেকে।
তো কলকাতায় উত্তরে আপনাদের জায়গা আছে, নাম হচ্ছে হিনমোটর ছিলেন।
মোটর, হ্যাঁ হ্যাঁ হ্যাঁ।
তো হিনমোটর আমার বন্ধু থাকে, তো ওখানে গেছিলাম, ওখান থেকে পরে গেছিলাম সোনারপুর আমার আরেক বন্ধু থাকে একটু।
আমার বাড়ি
ও তাই, আপনার মানে একটু সাউথ ইস্টে, কলকাতা সেন্টার থেকে।
আপনি ভালো জানেন কোথায় ম্যাপ, আমি অত জানি না বাংলাদেশে।
হ্যাঁ,
বাংলাদেশ অনেক কথা শুনেছি, ওই কেন কি আমাদের প্যারেন্টসরা তো বাংলাদেশ, আমার বাবা হচ্ছে কুমিল্লা, মা ময়মনসিংহ। যদি correct করে।
So we are most welcome to visit in Mymensingh University, everything, Jan Plasm Center, everything. If we get a scope in the future, along with Muntasir, or okay, you can make a plan sometimes.
অনেক গল্প শুনেছি ছোটবেলা, যে এখনো শুনি, মানে সবসময় যে খাওয়া দাওয়ার ব্যাপারটাই এত এত সুন্দর জিনিসপত্র আছে, খাওয়া দাওয়াটাই মোস্টলি হ্যাঁ ইন্টারেস্টেড। ইলিশ হোক ওইসব, এখন আমরা দায়িত্ব দিচ্ছি এসব নিয়ে, ওটা তো মেটাবলিজমের পার্ট।
আপনি দেখুন দাদা, আপনি কি করতে পারেন, কারণ এখানে অনেক কিছু করার আছে সিলেগেন্স। আমি যখন বাংলাদেশে যাই, আমার মানে মানে কত কিছু যে করা যাবে, এবং আমি অবশ্যই আমি নিজে অবশ্যই করব, কিন্তু আপনার 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 উপরে যে এখন সাইনবোর্ড আছে, আপনি যে সাথে যুক্ত, ওরা হয়তো মানে ওইটার যে যে কোলাবোরেশন টাইপের কিছু হলে ওটা একটা এক্সপোজার হতো আর কি আমাদের জন্য, সেটা ভালো। ইন্ডিয়া কিন্তু অনেক এগিয়ে গেছে। ইন্ডিয়ার আপনি দেখলেন এই কনফারেন্সে যেসব মানুষ আসলো, আপনার কবিতা বাবু এবং ওনারা আসলেন ইন্ডিয়া থেকে, আপনার আইএস, আইএসসি, আইআইটি। তো সেসব যদি বাংলাদেশেও কিছুটা হয়, তাহলে আমার মনে হয় অনেক কন্ট্রিবিউশন সিলিগেন্স রিসার্চে করতে পারবে। এবং ওইটা কিন্তু আমার একটা ওপেন কোশ্চেন ছিল অন দ্য ওপেন ফোরাম অন স্টাক 21, আপনি দেখেন কি আমি জানি না। তো আপনার যে প্রফেসর
মিরিয়ামই তো ওটাতে জবাব দিল, মিরিয়াম।
So it was very nice, very kind of her actually, in that she actually reached out. So and then one thing I'd like to another. So thank you for your time, Dr. Guha.
And
ফিলিংস এর জন্য থ্যাঙ্কস। এবং আমি যেটা একমত আপনাদের দুজনের সঙ্গে, যে আপনাদের যে একটা আন্তরিকতা ওইখান থেকে আগে আসা, আর আমাদের যেখানে একটা ইউনিভার্সিটি প্রতিষ্ঠান, এখানে যদি আমরা ধরুন এস্টাবলিশ করতে পারি, তো সবাই সব দিকে সেন্টার করে হোক বা যেভাবে আপনারা চান, আমরা কিন্তু সবাই মানে আমাদের ছেলেপেলেরা যারা আছে বা আমরা যারা আছে, আমরা আগাতে পারবো আস্তে আস্তে।
at the end of it's all about opportunity, right? সবাই যেখানে আসছে বা কিছু অপরচুনিটি পেয়েছে তাই বলে, নালে তো অপরচুনিটি আর যদি নাও দেখতে পারে লোকেরা যে কি করতে পারা যায়, মানে সেটাও খুব ইম্পর্টেন্ট, রাইট? লাইক তাই বলে ওই অপরচুনিটি ক্রিয়েট করাটাও, I mean, as a scientist, we always think about bench, right? এটা যদি যতটা ক্রিয়েট করা যেতে পারে, ততটা আপনাদের জন্য not only ভালো, it's for the students, it's for the future, right? Like both in the purview of Neuroplant, maybe there might be limited, but we can help you really set up a worm lab. Like I have experience in setting up worm labs from scratch, and I can bring in আমি আমার বন্ধুদেরও আনতে পারি যারা এখানে আছে, বা like collaborators would be very happy to set up things for you guys, like and as I said, like যেটা বললাম যে community-টা খুবই helpful. Not only that, there are other things also, যে aspects গুলো আনতে পারে, সেটাতে সবারই লাভ, like it's a win-win, right? Like it's, as I said, all about opportunity. The more opportunity you get, অত ট্যালেন্ট বাইরে আসবে, যদি অপরচুনিটি না হয় তাহলে ট্যালেন্ট কোথা থেকে আসবে, সেটাই ব্যাপার।
আপনাদের দুজনকে অশেষ ধন্যবাদ। আপনারা যে ইনিশিয়েটিভ নিয়েছেন বাংলাদেশের জন্য, সেটা অনেক হেল্পফুল এবং এটা অনেক দূর এগিয়ে নিয়ে যাবে আর কি এই ফিল্ডে। আমরা যেটা দেখি যে সেল কালচার নিয়ে যে আমাদের এখন আলাপ হচ্ছে, এই জিনিসগুলো প্রায় 50 দশক আগে, অনেক আগে শুরু হয়ে গেছে অন্যান্য দেশে বা ইন্ডিয়াতে। তো আমাদের দেশে সেটা শুরু হয় নাই। আপনারা যে জিনিসটা চিন্তা করেছেন যে এটা শুরু করা এবং এইদিকে একটা এগিয়ে যাওয়া, এটা অত্যন্ত আনন্দদায়ক এবং উৎসাহব্যঞ্জক। এখানে যারা স্টুডেন্ট আছে তারা কিন্তু সেটা উৎসাহিত হয় যে একটা পথ প্রদর্শক যদি থাকে, যদি থাকে, তাহলে একটু যদি নলেজের এক্সচেঞ্জ করা যায়, তাহলে অনেক বেশি কিছু করা সম্ভব।
সেটা আপনারা বললেন আপনাদের ওখানে যা ফ্যাসিলিটি আছে এখন সেগুলো যত মানে ওটা আপনারা আলাদা, আমাদের পার্সপেক্টিভটা শুনলে আপনারা আলাদা ভাবে ইউজ করো, এটা তো আমরাও করতে পারি এখানে, এটা আমরা এখানে করতে পারি, আমরাও জানতে পারবো, রাইট? আমরাও এটা এখানে করতে পারি, রাইট? তো এটা ল্যাটারাল ফ্লো, এটা বেসিক্যালি ওই না হচ্ছে একদিকে এমনি না যে আমি স্ট্যানফোর্ডে আছি তার মানে বিশাল বড়, এখানেও সায়েন্টিস্ট এমনি, বাংলাদেশেও ইটস অল অ্যাবাউট অপরচুনিটি। এখানে আমাদের কাছে বোধহয় একটু ভালো ইকুইপমেন্ট আছে, সেটা যা শুনলাম আপনাদের কাছে যে সবকিছু আছে, ব্যাপারটা হচ্ছে ওই আপনারা যত দেখবেন যত অ্যাক্সেস পাবেন তত নলেজটা বাড়বে, রাইট? তো সেটাই হচ্ছে বক্তব্য আমার, মানে
যেটা সি লেভেলে যে মেরিনের যে আপনার এক্সট্রাক্ট গুলো, মেরিনের যে আপনার ফ্লোরা গুলো এগুলো এখানে হচ্ছে এখন কাজ
স্ট্যানফোর্ডে আমি চিনি একজন প্রফেসর আমাদের টিমেই আছেন, লরেন ও'কনেল, তো উনি এসব আইসোলেট করেন আর ওইসব করেন, কিন্তু হ্যাঁ ব্যাপারটা হচ্ছে ওগুলো আস্তে আস্তে লাইক দিস আর দ্য স্টেপস, লাইক নেক্সট স্টেপস,
অপরচুনিটি আছে যে এখানে
there are always opportunities, like যারা আস্তে আস্তে ইন্টারেস্টেড হবে, বিকজ ফার্স্ট ইউ হ্যাভ টু শো দ্য মোমেন্টাম, রাইট? লাইক মোমেন্টামটা বিল্ড হলেই আস্তে আস্তে এগুলো আসবে, ইন্টারেস্ট আসবে, কিন্তু অনেক সময় মোমেন্ট না থাকলেই মোমেন্টামটাই হওয়া ইম্পর্টেন্ট, রাইট? এন্ড যেটা বলছি এখানের থেকে যদি না হয়, there are other ways also to build collaborations and all.
এটা অ্যাকনলেজ করতে হবে যে মুনতাসির ভাই 2018 তে এসে প্রথম যখন এলিগেন্স নিয়ে এখানে প্রেজেন্টেশন দিলেন সিভাসুতে, সবাই একটু এক্সাইটেড হলো যে না এটা করা যায়, এটা অল্প খরচে করা যায়, এটা
অনেক মানে বিশাল অল্প, মানে আমাদের তো আমি বলছি এত মজার ব্যাপার, আমি যখন সুইজারল্যান্ডে গেছিলাম তখন আমি নিজের গ্রান্ট নিয়ে গেছিলাম, আমি তো অবাক, আমি ওখানে সব ডিআইওয়াই করছি, তো আমরা বেসিক্যালি ওখানে আমার বস যে ও তো ইনকিউবেটর না কিনে ওর পয়সা পাচ্ছে ওয়াইন কুলার যেটা থাকে, মানে ওয়াইন কুলারও তো একই টেম্পারেচার, মানে ওটা ইনকিউবেটর হিসেবে ইউজ করা হচ্ছে। মানে আজকাল জিনিসপত্রের দাম, মানে এই সায়েন্স ডাজ নট হ্যাভ টু বি এক্সপেন্সিভ, আপনি সবকিছু সস্তা দামে করতে পারেন, ইট ইজ মিসকনসেপশন, লাইক আর সবকিছু ইবেতেও পাওয়া যায়, আমি তো আগেও জানতাম না ইবেতেও সস্তা জিনিস পাওয়া যায়, তো অনেক জিনিস শিখেছি এইভাবে ডিআইওয়াই করা যায়। কিন্তু সরি আমি আপনাকে কাটলাম, মানে আপনি কিছু বলছিলেন, আমি বাস এক্সাইটেড হয়ে গেছিলাম এলিগেন্স ল্যাব কত সস্তাতে।
এই জিনিসগুলো যে এক্সচেঞ্জটা, যেমন আমি যখন বাইরে গেলাম ওখানকেও তারা এই জিনিসগুলো কাজ করছে, খুব সিম্পল জিনিস, আমরা সেই জিনিসগুলো দেশে রেপ্লিকেট করতে পারছি না বা করার সেই আগ্রহটা অনেক সময় থাকে না, কিন্তু আপনার এই যে মুনতাসির ভাই এবং আপনারা যে জিনিসটা করতে যাচ্ছেন এটা আমাদের দেশের জন্য অনেক উপকার বয়ে আনবে, সেটা আমি মনে বিশ্বাস করি।
So thank you, Dr. Aziz. So Dr. Guha, so I think I will give maybe like one whole week, so maybe the week after, maybe if you would like to share some slides and maybe discuss with you.
আমি আমার স্কেডিউলটা দেখছি, লাইক উইক আফটার আই থিংক ইট শুড বি ফাইন, week after বলছো mid of July, right? Or mid of July, I think let me see quickly, but এমন করতে পারি আমরা, you have ওটা doodle poll বা when to meet, সেটা একটা পোল সবাই যারা আছেন এখানে সবাইকে, and we can also start a Google Drive. I think I've seen that is very helpful, like where you can share some papers from your work, we can share some papers and presentation from our work, that way like you have like এটা ট্রান্সপারেন্ট হবে, আপনারা দেখতেও পাবেন, আপনারা এডিটও করতে পারবেন, right? That way like it's not as if like I mean আমরা যা বলছি like sometimes it gets lost, তো ওখানে গুগল ড্রাইভে কি হয় এটা সুবিধা হয়, like everybody can share, edit, add things, and accessibility, right? Accessibility. So let's start with like maybe a when to meet, like and then I will also, I mean when to meet is I think free. তুমি না জানলে মুনতাজির আমি আজকে এখন আমাকে বেরাতে হবে, কিন্তু I can catch up with you tomorrow really quickly and we can make a plan, right? Like about this কালকে বা like I mean and we can share this like what the next steps are and all that, so that way we can involve all these folks, because it looks like it's a big community that you're trying to get, right? So a when to meet, like get a poll and then like when it works best for everybody, we should shoot for that, like, you know, so that way, like, you know, the more audience we get, the better it is, right? So let's start the poll and let's get this ball rolling. And we are really excited if we could help and bring others on board also. We have to
just one more thing. আপনি কার নাম বললেন যেন, যে মেরিন প্ল্যান্ট নিয়ে কাজ করে আপনার গ্রুপে?
আমাদের গ্রুপে মানে একজন প্রফেসর আছে স্ট্যানফোর্ড, লরেন ও'কনেল। I can share her thing in the chat. উনি
great. Because we do some marine stuff also in our group, like not only from plants we get the endophytes, we also get them from the marine sponges.
ওর ল্যাবে এক দুজন করছে, and ও basically ওই ব্যাঙের থেকে মানে ফ্রগের থেকে she isolates a lot of compounds, but she's also looking, and she's a young professor and she is amazing. এটা পাঠিয়ে দিয়েছি চ্যাটে.
Okay, yeah, thank you.
And yeah, I mean, the goal is, like, you know, we should set up a lab in Bangladesh for worms. There should be a lab everywhere in the world for worms, right? The worms are easy to use and like do things. So that would be an amazing thing to see, and happy to help in any way. Okay, I think this is it. Like, it was, I think so. Sorry, I'm like cutting you guys off. Like, আর কিছু বলার থাকলে বা কিছু ডিসকাস করার থাকলে, I'm happy to stay for another 5 minutes, but like, let me know, like, you know, if this is it, we can wrap it up today.
Hello, may I have a chance?
Yeah, sure.
Just to thank all of you. So thank you very much. And it is already midnight here, so I am hearing from all of you, and it's very nice talk. And especially I want to thank Dr. Sujoy Guha and Muntasir Kamal. আর অনেকক্ষণ থেকে বাংলায় কথা হচ্ছে, আমি বাংলা ইংরেজি মিক্স করেই বলি। আর সুজয় গুহা, ডক্টর সুজয়, আপনার বাংলা কিন্তু অনেক সুন্দর। প্রথমে বলেছেন যে আপনি বাংলা বলতে ভালো পারেন না, কিন্তু খুব সুন্দর বাংলা হচ্ছে আসলে। So I am one of the team members from Professor Hassan Nurjaman Talukdar sir. He is my direct teacher, and I am also working as the associate professor in the Department of Parasitology, Bangladesh Agricultural University. So in 2011, I joined a lecturer in this university, and currently we are working together, but sorry, time is running out. I will not stay long. Just I did my PhD from Hokkaido University, Japan, and my works are mainly on molecular diagnostics of protozoan parasites, mainly the leishmaniasis. And then I have a chance to work on cell-free DNA, and I did some high throughput sequencing analysis too, and I have publications on this. Maybe later on we can share. As you said, we can make a Google Drive and we can share our thoughts and research ideas and what we are doing right now. So as my professor said, that we have a very good, you know, highly motivated and hardworking people here. So any kind of collaboration is always welcome to us. And I think we can accept any case, I mean, we can establish C. elegans. And you know, in the last day before yesterday, Muntasir Kamal presented to us about C. elegans, and it seems very interesting to me. And thank you to him. And I think it's very much possible here in our lab also to build up a culture for C. elegans culture, and there are other techniques also. I will not say that we have much rich technologies here, but we can. In near future, we can establish, I believe, and hopefully we can. So thank you very much for giving us the opportunity to hear from you, and really I am learning a lot. And even from Bangladesh, today I have you know, known many of Bengali people who are doing excellent works as I have here. So thank you so much. Today I will not say a lot. Thank you very much to all of you. Thank you.
Dr. Akhtar, it is a mutual learning, right? It is not like, you know, it is like mutual. আপনারা যা অনেক কিছু বলেছেন, আমি অনেক কিছু শিখতে পেরেছি, জানতে পেরেছি, যেটা আমি জানতাম না when I walked into the meeting. So
thank you very much.
Take it as like a lateral flow, right? Like, you know, everybody is welcome to contribute and
exactly.
learn.
Exactly. Thank you.
কোনো ব্যাপার, এটাই this is how science works, right? It is a collaborative process. So we should do it. And yeah, way we can help. Yep. So yeah, thank you all. And আমাকে শুরুই বলুন, like আমার ডাক্তার এখানে মানে কেউ যদি ডাক্তার গুহ বলে কেমন অদ্ভুত লাগে, কেউ বলছে কে
আসলে কি বলে ডাকবো মানে বুঝতে পারছি না। মানে আমরা জানি যে ওই ডেভেলপ কান্ট্রিগুলোতে হয় স্যার স্যার বলারও, আমরা যেমন সবসময় সবাইকে স্যার বলি
না, একদম বাবা এখানে স্যার মানে
মুনতাসির ভাই, আমি কিন্তু ভাইয়াই বলে এসেছি, মনে হয় আমাদের এজও প্রায় কাছাকাছি হবে।
না মুনতাসির আমায় যখন ডাক্তার গুহ করে বলছে, আমরা কে ডাক্তার গুহ, এখানে তো আমি জানিও না, এখানে কেউ বলেই না, আপনার তো জানি, তো সেটাই অনেকটা অদ্ভুত লাগে আমাদের জন্য যখন ওটা বলে। আর এখানে এটাই ইনফর্মাল, রাইট? লাইক that's how it should be, right? Like when those things come in, that's when you don't like barrier যখন একটা থাকে, like those titles are great, but this also creates barrier, in my opinion. That's my opinion, right? Like we don't express our opinions freely. When I see like oh my god, like a professor আমি তখন একটা barrier create করিনি, আমাকে একটু লজ্জা হয়ে যায়, right? Like oh my god, I don't want to express ourselves. And this is what you would see, at least the culture in Stanford, যেটা আপনি দেখবেন, everybody is like free to share ideas. No question is stupid. Answers can be stupid, but no questions is stupid. Like, so it could come from anywhere, come from high schooler, come from a 6th grader, 3rd grader, anything. তো সেটাই মানে ওই ব্যারিয়ার গুলো রাখলেই গ্রোথটা হয় না, at least এটা আমি মানি। So we are all kind of equal here, so that should be great. Yeah.
তাহলে আমি আপনার সাথে কালকে পরশু আমি যোগাযোগ করবো আর কি।
নিশ্চয়ই আমাকে ইমেল করুন কালকে বা পরশু, we can quickly meet and set up the logistics for the next steps. That sounds good. Okay, thank you guys, thank you so much, nice to talk to you. Yes,
welcome, and thank you too.
Good night.
Late point.
Thank you.
Take care. Bye.
See you.
Bye.
See you.